Yeah, you're not the only one who's a little bit confused. My photograph, my title, and my abstract have been on the website for the conference since Easter, but somehow we didn't manage to make it into the book or the agenda, and I didn't even get a real badge. So, along with my jet lag, I'm beginning to doubt my own existence. So, but I will carry on anyway. Just to tell you a little bit about my background, is I spent 15 years working in the pharmaceutical industry as a computational medicinal chemist. I worked for companies such as Organon, Schering-Plough, Merck. And for the last couple of years, I've been the director at a new Dutch institute based in Amsterdam called the Netherlands eScience Center. And that center has two goals. It's to help scientists from a range of backgrounds, from a range of different from disciplines, climate research, astronomy, deal with big data and deal with the increasing amount of data they have available. And it's also about helping the same kind of scientists make use of advanced computing techniques so we can all increase the speed of our computation like Istvan did with the example he showed of using the GPUs. But I would like to talk a little bit about some of the things we learned and some of the things we did in the when I was still in the pharmaceutical industry, but also how that relates to these kind of topics. So the premise of my talk is that in the pharmaceutical industry, particularly in the last 20 years, we've really industrialized the way we generate data through things like high-throughput screening, miniaturization, parallelization of experiments. But what we haven't done nearly so well is keep up with the analysis and the dissemination of that data. And what I'd like to talk a little bit about is why that's causing problems and how some of these issues can be addressed. So if we look at science, and this comes from my kind of broader perspective now, working in a broader institute, if we look at how science is changing, scientists are collaborating much more, and they're collaborating in cross-disciplinary teams. They're also collaborating across different locations and different countries, etc., etc. We also see that there's an increase in the scale and the complexity of the projects they're trying to undertake together. And of course that results in larger amounts of data being generated and the challenge to somehow identify and extract the meaningful information within that, which is what we heard during one of the keynotes this morning about the importance of extracting that information and those hidden patterns. We also see that there's been, particularly in the last 5 to 10 years, a massive growth in the amount of public and externally curated data. So whilst a lot of what I talk about relates to the kind of industrialization of pharmaceutical research, this increase in data is also affecting everyone in academia as well, because they now have access to these kind of resources, and well, PubChem and Chembl are listed here, but there's been a progression over the last 10, 15 years in the generation of these kind of resources and and the challenge is how do you make use of them. And when I make use of, again, I come back to the keynote from earlier today, that means how do you use that to make better decisions. And finally, the other thing that none of us can escape is the digital revolution. That's changing the way all of us work. And in terms of chemistry, I think it's fair to say that the chemists have found it difficult to keep up with this digital revolution. And if you don't believe me, there were plenty of reports written recently about the way this is changing the way chemists have to work and some of the real challenges. And there's one report from, I think, earlier this year explaining about that chemists need to have more support in terms of data management, more support in terms of keeping up with the literature, and particularly chemists weren't adept at sharing their results electronically in the way that some of the other disciplines, scientific disciplines, have been. So big data. Normally, I'm glad I'm not the first person to mention big data today. I'm not even the second person. And big data refers to this increasing amount of data. And 5 years ago, we kind of talked about this data tsunami. This huge amount of data was going to come and wash over us and destroy us and prevent us from doing any work. Thankfully, we've moved on a little bit from that to now data's become an oil. It's become a resource. It's become something that you could harness, and if you can find the right ways of making use of it, you can turn it into something productive and something useful. So the challenge is how we get past, big data is a hype term, there's no doubt about that, but it is also an opportunity to look at all of our data practices and the way that we manage data and we make use of data, and particularly in that decision making. When we talk about data, big data, we talk about the 5 V's of big data. And some of them are more or less relevant to medicinal chemistry. In terms of volume, well, I work with an astronomy project that's generating, when they have the telescope on, they're generating about 50 DVDs of data per second. In that kind of context, the amount of data that was available to us in the pharmaceutical and in medicinal chemistry is, of course, trivial. So that's not our problem. Our problem comes with some of the other V's of big data. It's the variety. It's how you make sure your DMPK data and your biochemical data and the data comes from all of your other assays and inputs are being used at the same time to facilitate decision making. It's about the velocity of that data. It's about making sure you're generating data at the speed that allows you to impact the design cycle. There's no value in generating more and more data if it's or make it available if it's becoming slower than decisions being made by the chemists anyway. Then it won't have an impact. It's also about the veracity of that data, which I mean, of course, is the quality of that data and how much confidence we can place in it. Are we just generating a lot of data which clouds our decisions, or is it we generating data of a sufficient quality that it can really impact what we're doing? And value, we spend a huge amount of money building high throughput screening facilities and investing in the new proteomics technologies, etc., etc. Are we also treating the data that comes from these resources as of sufficient value that we're actually going to make use of it? In academia, and I know from my new experiences, it's much easier to go and buy a new large machine than it is to spend literally millions of dollars on that, than it is to spend the tens of thousands of dollars needed to support that with the right ICT. We have to treat the data we generate with value, otherwise we won't be using it to make the decisions we need to. So when I talk about data-driven drug design, I always get a little bit nervous on how the audience is going to react, because we all believe that what we're doing is data-driven. We all believe that we are taking the best information that we have available and using that to design our compounds. And the reality is, if you look at any real study on this, what we do is, in terms of medicinal chemistry, we focus on many other things. We focus on synthetic E's. We focus on, our favourite chemistry. The PhD chemistry comes up and over again. Chemical tuition, whatever that means to each of you. And particularly, we generate lots of chemistry dogma. These are the things that unfortunately drive too much of our medicinal chemistry, not really pure data-driven. This is what I know, this is what I don't know, and this is how I can use that, I can impact there. So what can we do to enable that data-driven drug discovery? And we should start at the beginning. And the first thing is ensuring that quality of that data. I led a team at Organon who were given the task to take a look at the quality of the data we were generating and see if we could find ways to improve it. And the biggest problem I found was this. There's 5 of these things here, sorry. This is a small pie chart of the accessing of the database at Organon where all of the data was collected. So we had a central repository where biochemical, chemical analytical, DMPK, cellular data was all kept in one place. Now the vast majority of that, maybe 95% of that data, was inputted by the molecular pharmacologists. This little green group here. This is who accesses that data once it's been put in the database. So the people who are generating the data hardly ever take a look at that data once it's been put into the database. The people who are making use of that data, of course, are the medicinal chemists and the computational medicinal chemists. It's not the people who generate the data. And there's a big dispatch between the people in the laboratories generating data and the people who have to use it. And that means that behind every data point there's kind of a story. In the industrialization of data, we just treat everything like it was made on a machine, therefore it's accurate, and we work with it, and that's fine. And it's not the case. And the pharmacologists understand that the data they're generating on a particular day may have been affected by something, or something happened, and all of that's lost because these people aren't the people who are making the decisions based on the data. And just a couple of examples. And if you're in academia and you're generating hundreds of compounds a year, this will seem completely incomprehensible to you. But if you're in the pharmaceutical industry, in companies making tens of hundreds of thousands of compounds a year, you understand how these mistakes happen. Just a compound here, compound number 1, it was measured twice in the primary assay. Once it was 30 nanomolar, and the next time it was 3 nanomolar. And of course, how that appears in the database is the average of the two. And that's the number that everyone sees in the database and uses and treats as if it's correct. Same with the next compound, big disparity here. Here we have compounds where the cellular data and the binding data are very different. This compound binds very well in a cellular assay, but apparently doesn't bind. The opposite is, of course, possible, where the compound is, not active in the cell but does bind. But why does that? What's happening here? And the fact is, this was just a compound, an example extracted from a database that had been there for 10 years. No one had said, what is going on here? Why is there such a disparity here? Because it's been industrialized, and there is no one's task it is to find these outliers. And again here, this is a reference compound, the compound that every compound was tested against. And over a 3-year period, its activity had ranged from 0.01 nanomolar to 0.66 nanomolar, 66-fold change in activity over time. And every other compound in that database was being compared to that compound. So what you understood about that, all those compounds, hundreds of thousands of compounds, came from some very flaky looking data, and this was just because the compound had come out of solution over time or it degraded in DMSO and so on. And none of these things are being picked up because of this industrialization of it without worrying about how to manage that. So the solutions for this are, well, first of all, you need good data repositories. You need to have a central location where all of the data generated in the project is collected. And that means all of it. I was amazed when I first started working with Merck, was that their equivalent to this, which still wasn't very good, didn't have any DMPK data, because the DMPK data was generated in a different laboratory, and the head of that laboratory didn't want to put their data in the database. So it wasn't there. So these drug designers were sitting there with half a database with the biochemical data and spreadsheets with the DMPK data. That can't be any way of working. So you need these central facilities, and you have to stop data being collected in drawers and on hard drives and on memory sticks and so on. And the worst problem for that is generating data externally, and people sending you a CD with data or a DVD with data, and then again put in a drawer because there's no facility for managing and storing that data. We spend hundreds of thousands of euros generating data in external companies and not worrying about how to manage and store that data in the long run. Once you've built these, people who do have these, and organisations, they tend to build them around this kind of chemical book kind of approach. Here's the compound, here's all the data I know about the compound, and then I can turn the page, go to the next compound, and see what I know about that compound. The problem with that is, well, first of all, the main problem is that you want to move to more of a kind of a spreadsheet kind of way, where you can browse data, where you're not worrying about what one compound is doing, you start thinking about what are the trends across all of my data. And the other problem with this is this tends to rely on you searching for, I'd like a compound who's with a nanomolar activity below 5. Well, what happens with the compound that has a nanomolar activity of 5.1? So you have to get rid of these kind of book-type approaches to storing data and move to spreadsheets. And in fact, spreadsheets are also already getting to the point where we've reached our limit with the size of our projects, where you have to start already going down to more abstracted visualizations of data, where you can start looking for trends across data and start looking for patterns rather than trying to look for individual compounds. And the reason why these databases are so important, because I did analysis. This is actually a picture from Organon, but I managed to do the same thing at Schering-Plough and Merck, and I got the same results. 40% of new projects came from old projects. So this, if you want to know where the new lead optimization project came from, the likelihood is it came from a smart pharmacologist or chemist saying, well, this compound over here, I would like to start a new project on this target. What do we have in the database? What do we already know? That's the most likely place to start. The best place to start from trying to design a new drug is from an old drug. That's just fact. And lots of other methods are important, but this is what you have. This is why you have to look after your legacy data and manage that, because that's where your new projects are going to come from. The other thing is, again, coming back to this idea of browsing data and moving to a more kind of visualization way. Today I've seen a lot of pictures that look something like this. I've only seen one or two pictures that look something like this, which what I mean is, instead of looking at individual compounds, you start looking at trends in data and trends across the data. For the simple reason, in a typical pharmaceutical project now, you're making 2,000 compounds. You can't look at 2,000 compounds individually, one by one. And if you can't look at them with all of the different properties in your head. You need to start changing this to a more data-centric approach. One of the things we did, we implemented Organon and went further, was a cybercarbon. We have one now in Amsterdam that we call the Collaboratorium, which is a terrible name. But it's basically somewhere where you have your data repository that everyone in a project team, in a drug design team, used to arrive in the morning, do the same thing, get a cup of coffee from the machine, and then open the data repository. Because that was where all the data generated the previous day was now available to them, and all the data generated over the previous 3 years or 5 years was available to them. When I first started working at Organon, we used to have project team meetings where pharmacologists, the medicinal chemists, myself, in vitro pharmacologists would come together. And the pharmacologist would hand each of us like a printout of data that they generated the previous 2 weeks in paper form. And he would give it to you with all of the data available. But it'd be the first time you'd seen it. And of course, there was 2 pharmacologists, an in vitro one and an in vivo one in the room. And they both had this data, and they both generated curves from this data. But they'd use different tools. So they had different numbers. So these 2 guys were over here shouting at each other because they didn't agree on whether it was 0.5 or 0.6 nanomolar activity. And the rest of us were just trying to pile through this to see what the hell is going on. That changed once we put the data repositories in place. But once you've got that and something to really look after the data, control the data, you also then need somewhere where you can discuss it, and the pharmacologist can explain to the medicinal chemist, this is what we're doing and this is what the result looks like, and then you can start making decisions as a team rather than people just sitting together in individual offices. Okay. The other thing that needs to happen, and I was very lucky to have lunch this afternoon with Victor. He was saying that medicinal chemists have to change, and they have to change a lot. And I'm not as brave as he is, so what I would say is we all have to become knowledge workers. All medicinal chemists have to become knowledge workers. That means they need to be able to prepare data, use statistical tools, analyse things, interpret statistical analysis. I wonder how many organic chemistry courses teach any of these skills at any serious level anymore. And my experience is there's very little of this done now, to the point where if I hear another biologist or chemist tell me, look at this graph, look at the statistic, look, it's a significant difference, and I ask what significant means, and they say, it's different. You can see it's different. We don't teach these skills anymore. We have to teach them if we want people to be able to make use of data. I'll skip on a couple of. The other thing that I'm really keen on, we discussed at lunchtime, was we've lost the ability to make decisions based on data. We based it on, again, chemical intuition and lots of other ideas. We're very bad at basing it on rational decisions and sticking by those decisions. And the example I have is being in projects where you're stuck in a maze, and you've made 100 compounds, you've made 1,000 compounds, and you're not getting anywhere. And what do? You just keep making compounds. And it's like being in a maze, hitting your head against a wall. At some point, you've got to take a step backwards. It might feel like you're going backwards, and it's a step backwards in the project, but that's the only way you're going to get out of the maze. And as scientists and medicinal chemists, we're terrible at doing this. We're terrible at taking a step back from what we're doing and saying, maybe there's another approach I need to follow. I skipped some of this anyway. And the other thing we have to learn to do is treat data dynamically. We're really good at looking at data, making a rule about it, and then deciding that's it, that's the rule forever. And we turn these into dogma. And just a quick example. The project team did an excellent job, really fantastic. They were making compounds, and they realised that these compounds, they were able to improve the binding, so they've gone from 35 nanomolar to 0.3 nanomolar. But that had no effect on the cellular activity. And of course, we all know what's going on. The compounds are too greasy, they're too lipophilic, they're not entering the cell. So they made a rule, and they said to themselves, well, I'm not going to make these compounds above 450, we're not going to make them with log P's above 4.5. Exactly what you want medicinal chemistry to do. Except that they never questioned that again for the length of the project. And they believed this for 5 years. And I was asked to go and have a look at this project after one of the mergers. We built a quick decision tree, and they were quite right. Don't make them too big, don't make them too greasy. Unless you have a pKa below, or you have a basic group there. If you had the basic group there, then you could make them quite much larger and much more greasy, and they would still have cellular activity. And they'd never taken a look at this, never thought about this. And what it meant was, well, I won't go through the analysis, because I'm running out of time, but basically there was a whole crop of compounds over here, based on the chemistry they've already done, so all accessible chemistry, that were, based on all of the work they've done, predicted really that they should be very potently active, and they were never going to make them because they'd made a dogma themselves that they didn't need to make, because they hadn't really looked at the data they were working on. So the other plea I have is that we improve our experimental design, and that means having statisticians back in early discovery. The pharmaceutical industry moved all of the statisticians to the clinic and to late development stages because that's where the money is, and we forgot that that's all based on something at the beginning. And if you start that badly, then it's all going to continue going badly. So get the statisticians back into discovery. And this is... sorry. Here's an example why. A simple project, it's a real project, an R group here and a second R group there. At the end of the project, I said to the medicinal chemists, what are the best groups here and what are the best groups there? And they gave me a list. 12 groups here, excluding a cyano, and I'll explain why over here, and 19 groups over here. That's 228 compounds. They'd actually only made 3 of those 228 compounds in the entire length of the project. And the reason was, and this is a project where they made 2,000 compounds, I should add. At a certain point, they decided the cyano, the nitrile group here, was the best group. So they decided to save themselves some time and energy in the synthesis, so they put the cyano on at the beginning of the synthesis, and then you could only change the R1 group. And that meant, all of the other, almost as good as the cyanos, were never made in conjunction with all the best of the R1 groups. And all of that information was completely lost. These aren't, if you put these chemists and these biologists into a room and told them to think about this, they'd solve these problems immediately, would never let this happen. But if you industrialise the way we try and make drugs, you tell people, stop thinking, just get on with it. And that's what you end up with. So Christ knows how many good compounds are in that 228. So, and finally, about this improved decision making. For a Rubik's Cube, there's something called a God's number. And what it means is, despite this, I can't even tell you what this number is. There's how many different configurations there are of a Rubik's Cube. But it doesn't matter which one of these positions you're in, you're never more than 20 steps away from solving a Rubik's Cube. And it's the same with the drugs. When I analysed Merck data and Shearing Cloud data and Organon data, you saw the same thing. We're making 2,000 compounds, but the difference between the first compound and the compound that goes into the clinic is actually less than 10 steps nearly always, and typically less than 5. So what happens is it takes you 2,000 compounds to work out, I replace this group with that group with that group, and then this group with that group, and then I've got my drug. And it takes you that long to find that out. And if you don't believe me, there's an article in Drug Discovery Today from 2 years ago where we show how similar the hits are to the eventual clinical candidates. So, I guess I'm up. Time up? Okay, then we'll go to the conclusions. So, the big data era is a big hype, and it is a terrible, horrible, overused term, but it is an opportunity for us to take a look at all of the way we treat data and we manage data. And I would just ask everyone that we start to give the data the value, at least the value that we actually spend generating it. We treat it like it's nothing after spending millions of dollars generating it. Please stop doing this. And we have to find better ways to make decisions based on that data. Because if you do, I think we can improve the attrition rates. So thank you very much. Sorry for running over.
I should have left my statistics points in my talk. Anybody question?
Yeah, I'm really impressed with what you've said. I remember.
Scared. Don't be impressed, be scared.
I've tried to use Zing curated database and Specs, Campbell, quite a number of them. In fact, I've used over 9 different databases. And the results have not been quite encouraging. The reason is because, but we were challenged to use them because enough publications are on them. You get what I'm saying now? So this is where I'm trying to come in. Some of the databases that we use, there are publications on them. And so we're encouraged to use them because we can easily quote them as references and so on and so forth like that. So I want to see, what do you say about this? Especially when, beautiful publications have been reported on this and then...
I know, okay. There's a difference between the quality, not the quality of the data. I mean, I think there's going to be a talk about PubChem tomorrow, I think, so I won't say anything nasty about PubChem. Not like, I don't have anything nasty to say. These are databases bringing together very disparate data generated in different laboratories at different times by different people. There is only so much quality you can expect from that, and the ability to be able to use that independently without thinking, it's not going to happen. I can tell you that, we have the same people. Well, I just showed you an example. The same laboratory, the same head of department, the same group leader, the people generating the data in the same assay, and they still have managed to get 66-fold spread in the data. So, the idea that the Campbell and PubChem and these are going to be able to fix this problem, and the data that you get from that, we're going to be able to build high-quality models on. You have to take into consideration those difficulties. Yeah, sure.
I don't know if it's a question or a comment, but first of all, I want to thank you for pointing out something that's been true for a long time, unfortunately. And the only comment that I want to make is that figures don't lie. It's the way that liars figure. And so the question is, are we lying to ourselves? That is the issue. We have all this data, but are we even thinking about it in the right way, or are we just lying to ourselves saying, because we have all this data, therefore we're brilliant?
No, we know what HTH data is. We know how reproducible it is. We know how reductionist these assays are. We know how they don't reflect the biology that we're trying to mimic. We know this. It doesn't mean they're not useful tools and they won't help us get to the solution we want to get to. But at some point we pretend to ourselves that these are reality, and they're not. They're a tool. That's all they are. They're an analytical tool. They don't say anything about biology.
You're absolutely right.
Scott, I think you will be very popular at the social hour after this session.
If I'm awake. I'm struggling a bit.
Let's thank again
Thank
to
you.
Scott.